Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
It's a pleasure to be here. And so this is going to be less modeling language and more model. But there's a notation for models in the background, and I think we've not been thinking that much in that direction, but it would be interesting. So what we're trying to do here is to model the problem of resource allocation. So the first thing I need to do is explain why it's important to model the cell. That's my short introduction. So there are three reasons. So first of all, systems biology is not actually that much about the system. We're modeling sub-processes, little cascades, MapK cascades, for example. We're thinking about them. And somehow we all know they function in the background of something, some bigger system. And surely they share resources. For example, all the things that are around have to be expressed, as biologists say. So they have to go through the successive stages of transcription, translation, and all sorts of intermediates. So the processes, people trust that their models are going to be, surviving the interference or the interconnectedness of the process as they are described with the rest of the system. So here, what we like to do is to describe the rest of the system, to give a rather simple, minimalistic model of what the context is. So, and context is a familiar notion in processes, right? As we understand them in computer science. So if we take an engineering view of the problem, obviously we're going to want to put circuits inside the system, and it's good if we can model somehow the relationship the circuit is having with the services it is drawing from the host, right? So we're talking about really microscopic embedded systems. And so for them, resources, energy, metabolites, all this is very important and key to their good functioning. And the third reason to do this, well, we can return to the first talk of this session. Okun was explaining how you can evolve circuits to perform better. But really, this powerful view, which comes from evolutionary biology, would be even better if it was informed by a model of the physiological, the global status of the system we're dealing with. So these are, three aspects of the reason to try to capture something of the global status of the system we're trying to manipulate or understand. So there are basically two ways to do it. One is to look at an exhaustive description, perhaps in a simple organism, of all the processes. And this is supposed to say two things. It's rather complex, although this is one of the simplest organisms known. It's a form of mycoplasma. And also to say that all the processes have to be seen together, and they're in this promiscuous environment sharing all sorts of things. So suddenly just modeling them separately is an approximation. So there's this exhaustive style of modeling, but there's also a more simple, sketchy way of modeling things, where you just have broad categories of processes and trying to look at the interconnectedness in a simplified manner. So we're going to follow that particular route. So I'll distinguish, for the sake of the argument, three different ways to do it. The first one is the one that was, for example, developed by Toysink and his co-authors, which is to be simple, tractable, but one disadvantage of this approach for us is that the problem of allocating resources, or the problem of how resources are drawn from how the various services, so to speak, of the system are directed to particular sub-processes, is solved by optimizing growth of the system. Well, this is a limitation because we know, or we think, or we'd like to have examples where growth or maximizing growth is not actually the goal which is pursued by the host, by the system itself, because maybe growth is not fitness, and fitness is what matters, or perhaps just simply the underlying mechanism that we're not describing are not capable of optimizing growth in all contexts. So to plug this as a principle precludes the ability to talk about those additional cases we're interested in. Then there's this very detailed model. So that is extremely interesting, and you can ask all sorts of questions, but it's a huge monolithic object, which is pretty much simulation only and very difficult to deal with. So we're going to try to reconcile the two things, get a mechanistic model where we don't postulate growth optimality, but at the same time stay simple. So I described the minimal model. So piece by piece. So it starts with S, which is some kind of abstract resource which is outside, and which you can import inside and obtain something called SI, so like sugar, internal sugar. And then that internal sugar in this abstract model is turned into a universal resource, which people sometimes call a precursor, which is going to be the one thing we use to build everything we need inside the cell to grow. So you can think of it as ATP, amino acid, everything, all the little bio bricks you need to put together a new copy of yourself. So now we need machinery to do this. So here we go. We have an enzyme T, which is going to import or transport what's outside inside, and then another enzyme E, which is going to transform what's inside into the precursor molecule. So that's good. Now we have also the need to build these machines. So now we have a dark blue material flow out of this precursor. We're now chaining them to construct big macromolecules, polymers, and we need a machine to do that. So that machine is R, like ribosome, and the machine is going to direct the flows of A's, and you see that the machine already has a choice between directed the one incoming resource to build either some E or some T. So we have allocation problems. Now this is biology, right? So it's completely autonomous. So we also need a machine to make a copy of the machine that's making copies of E and T, right? So ribosomes actually can make copies of themselves, which is quite convenient. So this is the third element we need. And we're done. We have a self-replicating system. But for the sake of the argument, we're going to distinguish two additional sectors in the economy. We can produce ETR, but also P, which is some generic bulk protein with no particular function, and a version of P, which is Q, which is self-repressing. And the interest of distinguishing between both is that we'll see that the behavior of self-repressing Q and of a non-regulated P is very different when the growth conditions change. The first one is going to be very sensitive to how fast the system is growing, which might be a problem, whereas the second one will manage to have a level of expression which is broadly invariant in lots of different growth conditions, which can be very handy. So the system is tractable. We have a bunch of variables, not that many reactions, not that many parameters, which is fortunate because we want to explore them. Each catalytic reaction, the little orange things here, is modeled by a Michaelis-Menten type of interaction. We also have reversible binding of ribosomes to messenger RNAs. So this is so this little arrow here is 2 stages: transcription, translation. But we won't really need to look into this. And finally, to keep things even simpler, we keep a deterministic version of all this and see what happens. So the problem is to understand how the resources here in synthesis, so the translational resources, if you want, are allocated towards the various things that this machine is supposed to be doing. So if you're the social planner of this little economy, you're going to have to decide how much of my resources do I push in this direction or that direction or that direction. And you have a kind of a dual problem, which is how you allocate your primary element material, your A, or ATP, or think of it as energy. So these are the two problems we'd like to understand. And of course, since everything is interconnected, some choices will have consequences on the rest of the system. So here are a few notations. R is a ribosome. Lambda is going to be the growth rate, so at the percentage per time unit at which the system is growing. Tx will denote transcription, Tl translation, and we have all these different 5 types of proteins. I arrange over them with the variable x. So the structure of the model is quite flexible. And actually, this kind of diagram, which we can call catalytic diagrams, which come from this paper, to my knowledge, maybe earlier, a very nice type of notation for this type of models. For example, here we have two types of inputs, and so we have two inputs to the metabolism, and we can ask different questions. We also have two types of enzymes, and we can ask, which is, so the allocation problem is refined, and you have more decision to make. So we can also talk about choice between two enzymes with the same diagram notation. Here, we can talk about the need to also take into account the replication of information, because actually that machine, the ribosome, does not make copies of something just after the something. It needs genetic information, which you yourself have to duplicate with a specialized polymerase, of which you can make new copies, using a gene which codes for the machine that makes copies of the information, so on and so forth. So this is a digression, but it's just to give you a feeling that it's a quite handy notation to talk about whole cell systems. All right, so I return to our model. So this is the proteic mass of the system. So let's look at what it's made of. So here you have the complexes that are made of a ribosome and a messenger of type X. So if I sum over these complexes and I multiply by the length of the ribosome, or the mass counted in amino acids, then I get the total number of amino acids that are actually involved into, that have been invested. It's my amino acid capital invested in making copies of something in the system. Then I've got the free parts, so these are the X's, including the ribosomes, that are not at work. So there's always a fraction of the systems where the machines are idle. So for each X, I can define the mass fraction of the corresponding protein. So it's just the total mass in amino acid divided by this complete mass of the system. And then the most key important definition here is the growth rate. So this is the total workforce at work. So these are the ribosomes actually actively translating something, whatever it is. And you multiply this by gamma of A. So gamma is the speed, the rate at which the ribosome machine is working in terms of amino acid per time unit. And it depends on the level of energy. So in a way that we can derive, and we have also some kind of Michaelis-Menten dependency. And then we divide this by total mass. So this is the idea that the growth of the system is measured relative to the current mass in terms of what's the percentage of increase of the mass that we get per reference time unit. So it's the growth rate in terms of how much biomass we produce relative to the present mass. There's another notion of growth rate, which is division rate, and here we postulate them to be equal. So we get now to fit that tractable model, and what we get is the data. So the data points are the little circles here. They're coming from a series of papers, and the model is figured by these little crosses here. So does that mean I'm 2 minutes? That's very short. So let's look at the points. So what we have here is, so if we follow the zeros here, we have the coordinates designate in the y-axis that the mass fraction of ribosomes, so the mass fraction of working amino acids, if you want, and the x-coordinate is the rate of growth. And when we move along the zeros here, we're improving the yield of the initial molecule injecting in the system. So we see that we discover a roughly linear and covariant relationship between how fast we grow and the amount of capital in the system that's invested into making new copies of itself, which is, depending on where you are coming from, quite intuitive or not. And here you can compare with the actual model points, and that they're quite close. So then when we move to 2 and then to 4 and to 8, what we do is we perturb the translational machinery. We actually decrease the efficiency of the gamma, the max gamma. So we impair the mechanism of translation. And again, we see a linear relationship here, both in the model and in the data points. And that linear relationship is now contravariant. So we see that whereas here we were, co-varying when we modified this yield of the initial molecule, here we're contravarying when we're impairing by using an antibiotic, we're impairing how well the ribosome, how fast the ribosome translates, just by occupying a little slot in the ribosomes that's getting in the way of doing translation. So I was going to pass for how many?
0 minutes.
I've got 0 minutes. Okay, so I'm not going to do that. And so these are two regularities that have been well observed and that the model recapitulates. And then there's another law that's been known since the 50s, which is called the growth law of Monod, which is how the growth rate depends on the amount of nutrient in the system, which we can also capture in this fashion. So I'll just finish with one slide, and we won't have time to show applications. Maybe I'll flash you some pictures. So the really interesting thing here, if you remember the choice we didn't make, which was to optimize for growth, is that there is no specification in the model that tells to the model how it should allocate those resources. It's entirely generated by the differential reaction of the transcriptional translational machinery to the level of energy in the system. So that may be just a tiny bit more concrete. So depending on the ATP level, the amount of, the normalized amount of transcriptional response we get will depend on the X. So if the X is the ribosomal machine itself, then you see that it rises later than the rest of the system. So this means that if I have more energy, then mechanically the entire economy will produce more of the final machine, and then we'll get this effect of shifting the fraction of the proteic mass of the system towards more ribosomes. So it's simple, but key example of the type of effect you get by the interconnectedness of the system. So I leave you with this as a meditation. So we have applications to synthetic biology. You can plug in a synthetic circuit and see how an oscillator, for example, relates to the growth conditions and how we can modulate the amplitudes, and depending on this, so we see effects. And another application is at the population level, the multi-scale design, where we see that we can explain critical decision-making in cell systems by the fact that there's a connectedness between different decisions being made by different cells that are mediated by the fact that they all draw from the same resource pool. So, and in fact, we established with the model that maximizing growth is not the same thing as being maximally fit in this fashion. Okay, I'll stop there.
Thanks, Vincent.